If you've been following health news, you know breast cancer isn't just one disease. It's a whole spectrum of molecular chaos.
That's right.
So for you, the curious learner, the challenge isn't just knowing the subtypes anymore. The real question is, how do we cut through all that complexity? How do we find the exact genomic fingerprint of an individual's tumor?
Exactly. We are definitely moving past that one-size-fits-all model.
Thank goodness.
To get to true precision oncology, what we really need is an unbiased, high-resolution map of the entire cancer genome. And that's what we're diving into today.
A massive study.
A massive study. It uses whole genome sequencing, or WGS.
WGS, okay.
And it was done on 1,364 clinically annotated breast cancers from the CUBRICS cohort.
And this specific cohort is, well, it's instantly compelling because it's so different from many of the genomic studies we've seen before.
It is. Most of them have focused on Western populations.
So what makes this Korean population study so unique?
It just provides a really necessary contrast. This cohort has a much younger median age.
How young?
Just 44 years old. And there are fewer cases of the most common type, the ER-positive luminal A tumors.
So that demographic difference gives us a completely different genomic snapshot.
A very valuable and distinct one, yes.
Okay, let's quickly define WGS for you at home. When people hear sequencing, they often think of targeted sequencing.
Right.
Which is kind of like searching for a lost key, but only looking under a single lamppost.
That's a great way to put it. Because WGS, by contrast, gives you the whole picture. It captures the entire spectrum of genomic changes.
So not just one or two genes.
Everything. The smallest typos, which we call single nucleotide variants, all the way up to the big catastrophic chromosome rearrangements.
The structural variants.
Structural variants, and also the physical amplification or loss of DNA segments, the copy number alterations. It's that high-density view that really maximizes our chances of finding new drivers and, more importantly, predictive biomarkers.
So, our mission today is to uncover the key insights from this huge analysis. What new cancer architects did they find? What kind of timeline did they map out for cancer's evolution? And I think most importantly, how do these WGS findings actually change how we approach treatment?
Well, the overview is pretty remarkable. They didn't just identify novel drivers. They showed that quantitative features you can get only from WGS.
Like what?
Like mutational patterns and tumor heterogeneity scores. These turned out to be surprisingly potent predictors of response to, well, everything, from standard chemo to targeted drugs like CDK46 inhibitors.
Okay, let's start with the scale of the genomic damage then. The researchers cataloged over 10 million somatic mutations across these 1,364 cases. That number reflects something we call the tumor mutational burden.
Or TMB.
TMB.
And TMB is essentially, the sheer volume of errors in a tumor's genome. The median TMB was high, but the concentration of that chaos was extreme in the one group in particular.
Which was?
The basal-like tumors, which often overlap with what you know as triple-negative breast cancers.
Ah, the really aggressive ones.
Exactly. That subtype had the highest median TMB by far. It shows this deep connection between a high level of genomic damage and, well, high tumor aggression.
So out of that just sea of errors, which ones are the actual architects, the drivers? They confirm the classics, right? Like TP53 and PIK3CA.
They did. Yeah. But the novel discoveries are where this cohort really shines. They found 4 new driver genes.
Okay.
And instead of just listing every name, I think it's better to focus on the type of disruption they cause.
Makes sense.
So 2 of these, BCL11B and RAB1, they're transcription factors. They act as master regulators, controlling how DNA is packaged and expressed.
So a mutation there is like taking the conductor out of an orchestra.
That's a perfect analogy. You're disrupting the most fundamental cell organization.
And what about the other 2, RAF1 and SPECI1? They were suggested to be tumor suppressors. That's a totally different mechanism.
A very different mechanism. If a driver mutation is like a stuck accelerator pedal, a tumor suppressor is the brake pedal.
Got it.
So truncating or disrupting those suppressors, even in a small group of patients, means the brakes are off. And that allows the cancer to progress.
Now, let's tie one of those classic drivers, TP53, back into this idea of chaos. When TP53 is mutated, it's linked to this profound instability.
Right. Specifically, higher ploidy, so extra sets of chromosomes, and a state we call homologous recombination deficiency, or HRD. We'll talk a lot more about that.
And this instability, this failure to repair DNA, that leads directly to another key concept, doesn't it?
It does. Intratumor genetic heterogeneity, or IGHE.
IGHE. And this is quantified by something called the MATH score.
The mutant allele tumor heterogeneity, or
Okay.
MATH score. Yes, a key finding from this WGS data.
So how should we understand the math score in a clinical sense? What does it tell us?
A high MAP score is, frankly, a terrifying sign.
Okay.
It signals highly active, late-stage mutagenesis. The tumor is frantically spinning out all these diverse, potent subclones. Think of a low math score tumor as a uniform army. It might be powerful, but you can target all its soldiers with one strategy.
Yeah.
And a high math score.
That's a highly diverse mercenary group. It's constantly adapting, finding new ways to resist whatever you throw at it.
And this measure of diversity, this math score, is it directly prognostic?
It is. High math scores were strongly associated with poorer overall survival,
Wow.
particularly in those already unstable TP53 mutant tumors. The message here is just crystal clear. The more heterogeneous the tumor, the harder it is to control.
So it just validates the power of WGS to quantify something that's subtle but absolutely critical for a patient's outcome.
Exactly.
Okay, let's do a quick recap. We've established that WGS on this unique CUBRICS cohort found novel drivers involved in chromatin remodeling and cell signaling. We know that basal-like cancers show the highest tumor mutational burden. And critically, the math score gives us a number, a quantitative measure of a tumor's internal diversity that directly predicts worse patient survival.
So now we can move from the drivers themselves to the footprints they leave behind. We call them mutational signatures.
Right, so if TMB is the quantity of mutations, the signature is the quality. Precisely. It's the unique pattern left by a specific cellular process or maybe an environmental exposure.
The study categorized these footprints. There are the clock-like signatures, right?
Yes, those are found in almost every single sample. They reflect that consistent background wear and tear over a lifetime.
But then you get the specific and I think really relevant signatures like homologous recombination deficiency, HRD.
HRD is one of the most critical clinical biomarkers that WGS can provide. It means there's a defect in the cell's main system for repairing double-strand DNA breaks.
And why is this so important for you as the listener to understand?
Well, because HRD tumors are specifically vulnerable to certain drugs.
Like PRP inhibitors.
PRP inhibitors and platinum agents, yes. It's a key to targeted therapy.
So what percentage of this cohort, this Korean cohort, was found to be HRD?
Using their combined signature markers, they found 23.1% of all cases were HRD.
So almost a quarter.
Almost a quarter. And this defect was massively concentrated in those aggressive basal-like cancers. Nearly 3/4 of those cases were HRD positive.
And WGS could pinpoint the cause.
It could. It found the usual suspects like BRCA1 and BRCA2 germline defects, but also new somatic causes, like a truncation in a gene called RAD51B.
We've seen what happens when DNA repair goes wrong inside the tumor cell. But what about the germline genetics, the inherited stuff unique to this population? The study pointed out a really striking difference in the APOB3A germline deletion.
This is a fascinating population genetics story. APOBEC signatures. They reflect DNA damage caused by some of our own cellular enzyme.
Okay.
And the APOBEC3B deletion, it's a common variant that can promote this hypermutation, was found in almost a third of the Korean cohort.
A third, so 31.8%. That's dramatically higher than the 8.5% we see in Europeans.
It is. So you have this highly prevalent variant that increases TMB in the people who carry it. But here's the surprise.
What's that?
Despite being so common, it was not substantially enriched in the breast cancer patients compared to healthy Koreans.
So it's not a strong predisposing factor?
Correct. It suggests that while carrying this deletion makes your cell's genomic environment more volatile, it isn't a strong cancer predisposing variant on its own, at least not in this population. It seems to require other factors to really kick off that full transformation.
Okay, let's shift to what I think is the true differentiator of WGS. Time.
Yes.
The study analyzed the timing of these huge genomic events, the long segmental copy number alterations, or CNAs.
These are the ones that involve whole chromosome arms. And this analysis, it provides an evolutionary timeline. It lets us date the cancer's history. And the finding is, well, it's profound.
What did they find?
These large recurrent CNAs are acquired incredibly early.
How early?
Around 20% of the tumor's total molecular timeline, which is estimated to be decades before the tumor is ever clinically diagnosed.
Decades? I mean, that just fundamentally changes the intervention timeline, doesn't it?
It completely changes it.
The genomic instability that sets the cancer on its path, like the amplification of the MYC oncogene, which the study links to poor survival, it begins when the patient is young.
Potentially during puberty, as another report has suggested.
So the full neoplastic transformation takes an incredibly long time. The cancer gets a multi-decade head start.
Exactly. And WGS allows us to visualize the genome, not just in 2D,
Location and mutation.
but in that critical third dimension, location, mutation, and time.
Okay, a second recap. WGS reveals these mutational footprints. HRD status, found in 23.1% of the cohort, is a key biomarker, especially in those basal-like cancers. We also noted the high prevalence of the APO-BES-APB deletion in the Korean cohort, though it doesn't seem to be a strong predisposing factor. But most critically, WGS analysis shows that genomic instability, these big CNAs, can start decades before diagnosis.
So beyond those small base pair changes, WGS really details the large-scale reorganization of the genome, what we call structural variants, or SVs.
The ones that physically reshuffle huge chunks of DNA.
Right. And these SVs, they lead to really important functional changes, particularly through translocations, and you highlighted one of them before, something called enhancer hijacking.
Let's talk about that, specifically around the ERBB2 locus.
So enhancers are like accelerator pedals for gene expression. And super enhancers, or SEs, are these powerful, dense clusters of accelerator pedals.
Okay.
A structural variant acts like a disruptive construction crew. It physically breaks the DNA, takes the super enhancer from its normal distant location, and translocates it right next to an oncogene.
Like ERBB2.
Like ERBB2.
And the result of moving that high-powered accelerator right next to the engine.
The tumors with these specific SVs showed significantly higher ERBB2 gene expression than you would ever expect, based only on the number of copies of the gene they had.
So it's a way to crank up the volume without having a huge copy number.
A very efficient way to do it, yes. This mechanism, this enhancer hijacking, is a critical way that SVs drive oncogenesis.
Let's shift to another powerful physical mechanism, extrachromosomal DNA, or xcDNA. This sounds incredibly important for gene amplification.
It is. It's the physical mechanism behind the highest levels of amplification we see. ecDNA are these small, circular DNA segments that exist outside of the main chromosomes.
And because they're separate.
They can be replicated independently and very, very rapidly. They are the key engine for what we call focal amplification.
How common is this mechanism for the most famous breast cancer oncogenes?
Incredibly common. The study found that more than 40% of the focal amplifications in major oncogenes like ERBB2, CCND1, FGFR1 were formed by xDNA.
Over 40%.
Yes. So this tells you that xDNA is not some rare occurrence. It's the primary physical mechanism these tumors use to get those high copy number gains that fuel their aggressive growth.
And finally, we should just touch on the recurrent fusion genes, where two different genes get spliced together.
Right. The study found fusions like CCDC170ESR1 in luminal cancers, which have been linked to endocrine resistance.
And because these are mostly clonal,
it suggests they likely play an early initiating role in the tumor's development.
Okay, a final recap for this segment. We've learned that structural variants can physically rearrange the genome. This leads to this sophisticated enhancer hijacking that dramatically boosts oncogene expression, especially for ERBB. And crucially, over 40% of those key focal amplifications are actually driven by extrachromosomal DNA, showing how cancer gets those high copy number gains.
So now let's bring all this genomic detail back to the bedside. The ultimate utility of WGS is predicting how a patient will respond to treatment.
And we can start with refining HER2 testing.
Exactly.
The traditional standard for HER2 is immunohistochemistry, IHC. It's sensitive, but maybe not precise enough. This study compared it to WGS-derived data for predicting a pathologic complete response, or PCR, after neoadjuvant therapy.
And they found that the absolute ERBB2 copy number threshold from WGS was the superior predictor. Specifically, a high absolute copy number 33 or more demonstrated significantly higher precision and specificity than the traditional IHC score.
So the power is in the absolute number.
It is. It's that quantitative measure. And it didn't matter if the amplification was linear or driven by xcDNA, WGS gives you a more precise number, and that number predicts response better.
Now, adding a layer of chaos to the prediction, they also found that chromothripsis, which is this catastrophic chromosomal shattering, was enriched in the cases that did achieve a complete response.
It was. And when you combine chromothripsis with the high ERBB2 copy number, the prediction got even better. So sometimes that genomic chaos can actually signal a vulnerability to certain drugs.
That leads to what might be the most significant clinical application of WGS, what you call the HRD dichotomy.
Yes.
The HRD status we've been talking about, it acts like a high-stakes two-sided coin. It gives you opposite predictions depending on the treatment.
This is such a critical distinction. Let's look at triple-negative breast cancer patients who are getting standard chemotherapy.
Okay.
Here, HRD status is a good sign. It predicts superior disease-free survival. The DNA-damaging chemo works really well because these tumors can't repair themselves.
But the outcome is the complete opposite in hormone receptor-positive cancers getting a specific targeted therapy.
Exactly. The CDK46 inhibitors plus endocrine therapy.
Right.
In that setting, a high HRD score predicts significantly worse progression-free survival. In fact, HRD was the single most significant factor for poor PFS, with a hazard ratio over 10.
Over 10, that's huge.
It is. It seems these highly unstable HRD tumors develop resistance much more rapidly to that specific targeted combination. This is why WGS is essential. Without it, you might be giving a highly unstable tumor the exact wrong therapy.
And as a final note, let's revisit that concept of genomic time and resistance. WGS provides that three-dimensional view, you said.
And this is a perfect illustration. The researchers tracked a patient who developed resistance to a PRP inhibitor called talazoparib. The patient had a pre-existing BRCA1 deletion that made the tumor HRD and therefore drug-sensitive.
So the drug should have worked.
Initially, it did. But post-therapy WGS sequencing revealed the tumor acquired a second small deletion in BRCA1.
And that second deletion somehow fixed the first one.
It restored the reading frame. The tumor cell effectively engineered itself. It converted from being homologous recombination deficient and sensitive back to homologous recombination proficient, resistant.
And they could date this event.
WGS allowed them to date this rescuing mutation to a precise point in the tumor's evolutionary timeline. It showed resistance emerging in real time in a subclone.
This entire deep dive just shows that WGS isn't just a catalog of mutations. It's this powerful tool that gives us a comprehensive, time-aware, and quantitative understanding of cancer's evolution.
Absolutely. The ability to visualize the genome in 3D location, mutation, and time is critical. It helps us understand not just how cancer evolves over decades, but how it bypasses treatment in real time.
So here's our final provocative question for you to mull over. Given that this genomic instability often starts decades before a tumor is even clinically detectable, and given that our key predictive biomarker, HRD, predicts completely opposite outcomes depending on the treatment you use,
How might future screening and prevention strategies need to fundamentally change? How can we target these genomic instabilities and that decade-long head start they provide long before the cancer is even visible?
a question that really redefines the entire timeline of cancer intervention. Thank you for joining us on the Deep Dive.